Hello students, we shall continue the lesson inheritance and variation. Now the characters are inherited through the chromosomes. So in this lesson we are going to study the structure of chromosomes. Chromosomes, they are filamentous in nature, found in the nucleus of the eukaryotic cell. Each chromosome is made up of DNA, histone and non-histone protein. The chromosome is made up of chromatin. Chromatin is a filamentous structure. It is wrapped by the double-stranded DNA, and the subunit of the chromosome is nucleosome. Nucleosome is made up of about 2 tons of DNA with 8 histone proteins. Now in the examination there can be a question. Write note on the chromosomal theory of inheritance. In 1900, three scientists, Hugo de Vries, Correns and Skermark, rediscovered Mendel's work on inheritance of traits. And due to the advancement of the microscopy, scientists were able to observe cell division. What are the cell division? Mitosis and meiosis. Mitosis occurs into the body cell, meiosis for the formation of the gamete cells like sperms and the eggs. Walter Sutton and Theodore Boveri studied the parallel behavior of the genes and chromosomes. Chromosomal theory of inheritance, popularly known as Sutton's theory, in which you can describe as chromosomes are always pairs in the somatic cells. Somatic cell is a body cell. During gamete formation, so in the meiosis, homologous chromosomes, they pair and segregate and they separate independently. Each gamete contains only one chromosome because they are separated from a pair. Nucleus of the gamete contains chromosomes which carry all hereditary traits. Chromosomes are the link between parents and offsprings. The fusion of haploid male gametes and haploid female gametes restore diploid number of chromosomes. So simply you can describe as chromosomes are found in pairs. During gamete formation, homologous chromosome pair and segregate. Each gamete contains only one chromosome from pair. Nucleus contains chromosomes. Chromosomes carry hereditary material. The fusion of haploid male gamete and haploid female gamete, that is sperm and egg, restore diploidy. Now the question is, describe the structure of the chromosome. See the structure of the chromosome. The chromosome, simply you can see, consists of the telomere, chromomere, chromonimeta, centromere, secondary constriction and satellite. So chromonimeta and chromomere. Chromonimeta means it is the double-stranded DNA, and here the chromomere, the chromomeres, these chromomeres, they are beaded appearance, and these chromomeres are the sub-unit of the chromatin for the expression of the genes. Telomere is the last portion. Centromere is the central portion, a disc-like structure. Secondary satellite or secondary constriction, that is centromere is the primary constriction. Secondary constriction, when secondary constriction is there, the chromosome is called to be the satellite chromosome. So chromosomes are filamentous bodies in eukaryotic nucleus. The term chromosome was coined by Walder. multiple choice question. Who coined the word chromosome? Waldeer. Size varies from 0 to 1 micron to 33 micron and breadth 0.2 microns to 2 microns in thickness. Chromosomes are made up of DNA, histone and non-histone proteins. Function of the chromosome to carry the heredity. What is heredity? Position of nose, eyebrows, color, height. Same to the plants. Ploidy. Ploidy designates the number of the chromosomes in their multiple number of the basic number. For example, monoploid, haploid, diploid. When there is no multiple of the number of the chromosome, it is called aneuploid. That is the addition or deletion of one or more chromosome. That is not the multiple of the original number like 2 to 4, 4 to 8, 8 to 16. Green structure shows here the chromosomes and the bodies are called to the sister chromatids. Chromosome is made up of the chromatids. Sister chromatids above, sister chromatids below. Chromosomes are visible at metaphase stage. Typical chromosome consists of two chromatids. They are joined at the centromere at primary constriction. Primary constriction consists of disc-shaped plate called kinetochore. Some chromosomes possess additional one or two constrictions called secondary constrictions. So they are called satellite chromosomes. Earlier I described the structure of the chromosome. In examination you can draw the simple structure and describe accordingly like telomere, chromomere, chromonemata, centromere, secondary constriction and satellite. This is the X chromosome having chromatin body, sister chromatids, then centromere with core of DNA. We are continuing structure. At secondary constriction, nucleolus become organized during interphase. So secondary constriction means it is nucleolus organizer. Satellite body is attached at secondary constriction. 2. Each chromatid contains long unbranched slender highly coiled DNA thread called chromonemata. What is chromonemata? It is a thread of the DNA extending through the length of the chromatid called chromonema. End of the chromosome is called telomere. Now in the examination question become describe the structure of the chromosome or describe the types of the chromosome. Types of the chromosomes are here. First is metacentric. In metacentric, centromere is at the center. It is generally V-shaped. Then submetacentric, it is not exactly centromere at the middle but near to the middle, so it is generally L-shaped. Acrocentric, near to the end, centromere placed, it is J-shaped. And telocentric, it is I-shaped, exactly at the end of the chromosome. Centromere is placed at the tip of the chromosome. So 4 types of the chromosome: metacentric, V-shaped, submetacentric, L-shaped, then acrocentric, J-shaped, and telocentric, I-shaped. Sex chromosomes. Now in the human there are 44 autosomes, means 22 pairs, and 1 pair of the sex chromosome. If there is XY combination, male child, XX combination, female child. So there are 2 sex chromosomes. They are also called to be the allosomes. X chromosome is longer than Y chromosome. X chromosome contains more genetic material than Y chromosome. X chromosome has more euchromatin and less heterochromatin. Y chromosome has more heterochromatin and less euchromatin. So this is the difference between the chromosome. Both the chromosome contains the homologous part and non-homologous part. Crossing over takes place in homologous region only, and as no crossing over in non-homologous chromosome, so the genes which are linked to each other, they are not separated and they are directly come into the gamete. See here in the picture, this is the X chromosome and this is the Y chromosome. Both have non-homologous region and homologous region. Homologous region means similar sequence of the genes. X chromosome is longer than Y chromosome. X chromosome contains more genetic material. Y chromosome contains less genetic material. X chromosome has more euchromatin. Y chromosome has more heterochromatin. Non-homologous part of the X chromosome is larger than Y chromosome. XX combination female child, XY combination male child. Crossing over. Crossing over is the important part for the evolution in heredity. Crossing over takes place during the meiosis. During the meiosis, there are phases like prophase, metaphase, anaphase and telophase. So prophase 1 is longer, in which you will find the stages: leptotin, zygotin, pakitin, diplotin, dikinesis. So the pakitin of the prophase 1 shows the crossing over, in which the homologous chromosome come near to each other and part of the chromosome is shared, that is the sharing of the genes takes place. So new combinations are coming to the newly formed gametes such as eggs and sperms. So recombination occurs during the pakitin of the prophase 1 of the meiosis. The term crossing over was coined by Morgan. Remember, crossing over term was coined by the Morgan. Crossing over comprises four steps like synapsis, tetrad formation, crossing over, terminalization. Recombination increases natural selection. So crossing over is a process that produces new combination or recombinations. It is new combination of genes by interchanging and exchanging of the corresponding segments between the non-sister chromatids of homologous chromosomes. What is the non-sister chromatids? When homologous chromosome pair, the chromatids of the two separate chromosomes are called non-sister chromatids. So there is an exchange of the segment of the genes. That is the crossing over, and due to the crossing over, there is a recombination in prophase 1 of the meiosis. It occurs through the pakitin of prophase 1. Then it comprises stages like synapsis, that is the pairing, tetrad formation, crossing over, terminalization. It increases natural selection and variation. So what types of the questions are going to come? Like describe the structure of the chromosome. What are the parts of the chromosomes? Centromere or primary constriction, secondary constriction, telomere, chromonemata and chromomeres. Then types of the chromosome: telocentric, acrocentric, submetacentric and metacentric. You have to remember the shapes like I, J, L, V. Then write note on the recombination, what is crossing over, and write the difference between the X chromosome and Y chromosome. Thank you.